Well, good afternoon, everyone. It's slightly afternoon, so it is afternoon. I want to welcome everyone who's tuning in online. We did have a nice session at the meeting here in Annapolis on harmful algal blooms, and we have three of our presenters have agreed to stay on for a roundtable discussion. We have gotten a lot of questions from from you guys, basically, that are out there in the cyberspace, and I'll be responsible for sort of consolidating them and posing them to our panel at the end. My name is Chris Jones. I'm at George Mason University, Potomac Environmental Research and Education Center. We're on title of Potomac. And I want to point out that there was a strategy behind this session and behind the roundtable, and that is that we're finding HABs throughout the watershed. We're finding HABs in flowing waters, we're finding HABs in tidal freshwater, and we're finding HABs in estuarine water. And as a corollary to the streams, we're also finding in the lakes in the watershed. So it's not something that's restricted to one part of the watershed. It's something that we will all need to be dealing with. I also want to point out that we have still developing techniques to both measure the strength of HABs, to measure the toxins and what generates toxins, what specific conditions may be associated with the generation of toxins, and also why and how the blooms are initiated. I think that's, and trying to do something to monitor bloom initiation. So that's just my overview of what we talked about this morning. So without further ado, each of our panelists will make a short 8-minute presentation summarizing their experiences and some of their data. And then after that, we will have a half hour for a questions that can be submitted on the chat now, but also many were submitted prior to that, and I'm busy consolidating those. So without further ado, our first presenter, Rosalina Kristova from George Mason.
Hello, everybody. My name is Rosalina. I'm from George Mason, and I'm going to talk about HABs in freshwaters. So in freshwater portions of tributaries to Potomac River, usually we have relatively shallow ecosystems which are receiving a lot of light. And in conditions like that in flowing water, usually we find the problematic algae on the bottom. We call them benthic cyanobacterial growths, and some of those may produce toxins. Particularly the neurotoxins, cyanotoxin A and saxitoxins, are kind of in consideration because they may have quite a negative effect on ecosystem and also domestic pets. Still, we don't know clearly which factors specifically control their distribution. We're in the process of studying their environmental variables and their preferences in the conditions which are promoting and supporting this type of growth. So far, there are studies which indicate that the water velocity, temperature, correlations between nitrogen and phosphorus nutrients are maybe very important, and they may determine the growth of Max. Thank you so much. And here on the image in the middle, you see our typical view of underwater view of these mats, which are attached. Some species grow attached to the substrate, some are not attached, and also we study them in laboratory cultures, which are indicated to the left. So why they are issued is benthic HABs. They pose environmental problems. They are toxic for aquatic organisms, and also they are reason for dog kills. The toxins which are produced may they have acute toxicity, particularly the neurotoxins. and often we cannot enjoy the beauty of the rivers, which are experiencing blooms. So recently, last year, we started study on Shenandoah River, which is experiencing, maybe historically, I would say, blooms, both of green algae and cyanobacteria. And the question is, what is growing when, and what is the duration of the blooms? So here, beautiful slide of algae. If you see something like that in the field, you should not be concerned. Everything that looks grass green, and it's quite large, it's most likely green, different type of green algal growths, particularly Cladophora. It's quite large filamentous alga, and also golden looking growths, which are very soft if you touch them. Although you always should be careful when you are touching algae, these are that and also harmless. But if you see something like that, blue-green, brownish attached to the substrate, it's maybe a consideration. So the major genera, I'm not going to go into scientific names here, but still some mats are really blue-green, or maybe a little bit brownish. These are different species of cyanobacteria which form films on mats. Some are attached to the substrates, like microcoleus. Some are not attached, they just lay on the surface of the sediment. They may produce toxins, we should be careful with those. Another important organism, which is the benthic cyanobacterium, which is forming very large mats. On the bottom, they are described as black hair material. They really look like that. It's something that we need to be careful when if we see something like that. Also, these organisms are photosynthetic. They are good in the sense they fix the carbon CO2 from the air and they produce oxygen. In these oxygen bubbles, when they trap into the mats, they elevate the mats on the surface of the water, so you may see them also on the top of the water. And here,
Yes.
cancel?
Yep, cancel that, please.
Doesn't, okay. And my last slide here is again about both genera Microcereus and Microcoleus. We already observed seasonality. So they grow, they produce different abundances during different seasons, and our studies are continuing. And this work was done with the help of many students. Thank you.
Okay, thank you, Dr. Christova. Our next speaker is Dr. Judy O'Neill from Maryland, University of Maryland.
Thanks so much. Thanks to everybody who's online. We've had a great session here today, and I'm just going to share a few slides about Toxis filamentous cyanobacteria that's growing on seagrasses at Susquehanna Flats, and I'll show it where that is in a moment. This is just our research team from the University of Maryland Center for Environmental Science, as well as folks from Maryland DNR, as well as St. Mary's College of Maryland. So our site is up here. Here's the Chesapeake Bay that we all know. And our study site is up in the tidal fresh, which is called oligohaline portion of the Chesapeake Bay, at the mouth of the largest tributary for Chesapeake Bay, which is the Susquehanna River. And in green here, you see the outline of the submerged aquatic vegetation, or SAV bed, that is the largest and most diverse bed in the Chesapeake Bay, very shallow. So interestingly enough, during the 70s, this SAV bed disappeared due to Hurricane Agnes, but it has responded due to enhanced water clarity during extended dry periods and long-term reductions in nutrient loading. And just when things are going well, in about 2004, we started seeing cyanobacteria blooms, which were identified as Lingbia, and Lingbia has now been reclassified as Microcereus, and Rosalinda just told you a little bit about it. So what is this Lingbia, now Microcereus? This is what it looks like macroscopically. As Rosalinda said, it's like black hair. In Australia, they call it mermaid's hair. I'm not sure I want to meet that mermaid, but anyway, this is what it looks like in the shallow flats growing and overtopping the seagrasses. And this is work from Cassie Gerbitz, who's one of our co-PIs from when she was a PhD student at Horn Point. And I don't know if you can see here, but this is from the center of the SAB bed to the outside of the SAB bed, and this is percent SAB cover. So it's 100% in the center. As you go to the sides, it gets down, gets lower, and that corresponds to the percent Lingbia cover. It's almost the same as the SAB, and that also is coincident with dissolved inorganic nitrogen, which is very low in the center, but higher on the outside. And so that seems slightly incongruous, but why is there more in the center? And that is because Lingbia, Microcera, is a nitrogen fixer, and nitrogen fixation is where these organisms probe certain bacteria and cyanobacteria, which used to be called blue-green algae, can break this very stable nitrogen bond of atmospheric nitrogen that's dissolved in the water with this enzyme nitrogenase for ammonium for their own use that can also get into the system once they decay. So we had questions about whether this nitrogen fixation could be seasonally a net nitrogen source in these systems rather than what we usually think of as a seagrass bed or SAB bed as being a sink. So we also wanted to know whether it was inhibiting the seagrass viability and recovery. So these are just some of the sites from DNR of where Lingbia blooms have occurred, mostly in the freshwater reaches and including the Potomac, which Rosalina alluded to. And so these were just some of our factors. We're just trying to understand, you know, why it's there, why it's growing, and whether it's going to interfere with the recovery of the SAB bed. So this is research that's funded by Maryland Sea Grant, and we have a three-pronged approach. We're doing field surveys on the flats. We're doing laboratory experiments, and hopefully eventually we'll get to some ecosystem modeling. I just thought I'd show you what it looks like. This is us. We do most of our sampling by snorkeling. We do surveys of SAB cover and percent Lingbia cover. And if you notice, this site doesn't have a lot of Lingbia on the bottom. This is a nice clean site. That's Brooke Landry from DNR. This site is like a little in a nice Vallisneria bed. And the Lingbia is still down on the benthos at the bottom, but you can see it's like a thick mat on the bottom. And you can see the sediment coming off of that. And we think that's really important in the ecology. It seems to be getting phosphorus off of the sediment. And here's a gunkier site, and this is Cassie Gerbitz from St. Mary's College, and she's taking a core in amongst the gunk. You can see it's actually covering the seagrass here. There's also some oscillatoria or formidium in there. It's not just Microcera, but it's mainly Microcera. So you can see it's kind of an interesting site. So why is it occurring in the center where there's low nitrogen? As I said, it's a nitrogen fixer, so it's stimulated by nitrogen fixation. And another thing we have found recently is that it produces saxitoxin, which is a toxin that is known to cause paralytic shellfish poisoning in other regions. We don't see that here, but we haven't investigated whether it gets into the food web or not, but we'd like to do some future research on that. The saxitoxin levels correlate with the Lingbia biomass and also negatively with ammonium because saxitoxin is very nitrogen rich. And so we think that's taking that up. And so these are questions that are still to be answered, whether it's impacting the food web, whether the toxin is transferred, are nutrients increasing toxin, is climate change increasing it, and are the cyanobacteria going to affect resilience of seagrass beds? And I think I will just leave it at that and thank Sea Grant. Thanks.
Okay, moving along, and we'll try to get this out of the way. Not out of the way. We'll try to make sure we save some time for questions Since we got a late start due to the technical issues. So the last of our three panelists is Martin Woolhall from ODU.
Okay, hey, and I've actually tried to look in the pre-question, so hopefully I answered some of those, and sorry for the people in the room, but I showed some of these slides just a little bit. But this is our sort of it takes a village approach to, or maybe a region, not just a village, approach to HABs in the main stem. We have had a lot of blooms, and they appear to have been increasing over the last 30 years in the Chesapeake Bay, being linked with nutrient over-enrichment and anthropogenic perturbations. Many of these are harmful, produce toxins, and we get taxa that bloom seasonally with some regularity. And I'll show you a picture of that in just a minute. We have some existing Virginia estuary monitoring programs and the Bay program modeling that's monitoring that happens monthly. Also occurs in Maryland, although the phytoplankton program has been discontinued, is my understanding. No, it's bad? Okay. And we have VDH shellfish sampling, as well as HAB response everywhere in the Bay. And we also have time series sampling and some time series stations we've established near points of bloom initiation in the lower Bay. This is a slide that just shows the seasonality of the blooms based on that data on the panel on your right as you're looking at the screen. We've sort of developed a climatology of when the blooms happen in the lower Bay, at least. And we think that these blooms have been increasing over time based on data. The left panel is from 1994 to 2006. The red dots are blooms of Margolophodinium polycricoides. In the center, the panel shows blooms from 2007 to 2014. And then on the far right is blooms from 2015 to 2022. And you can see that the Margolophodinium blooms have increased and Alexandrian monoletum blooms have emerged subsequent to these blooms. And we don't know if we're opening up new niches with climate change and rising temperatures. But these blooms can be spectacular, and they reach cell densities of thousands or hundreds of thousands per milliliter, and they make the news because people take real notice, especially when they bioluminesce. This is just a picture showing the patchiness of blooms, and the picture on the lower left is just showing chlorophyll, just showing the water and the chlorophyll concentration at different times of the day. So even over the course of the day, these species vertically migrate, and the water can in turn deep mahogany over time. We have developed a conceptual model of this bloom, and it essentially looks like a growth curve. And while most people are making measurements once a bloom is developed, well developed, the $1 million question is why they initiate. And so we've been doing, and so with our time series site, we've established that we can actually detect blooms better when we monitor more frequently. At the bottom, the gray dots are the Chesapeake Bay program climatologies, and the other colored dots are different years where we've monitored season almost daily at our time series sites where bloom develop. We've, based on our conceptual model, we figured out a lot of different holes we need to plug in order to bring these models, and the different meteorological forcings, physical forcings, biological factors. And with addressing some of these questions, we're trying to build actual models of the bloom. And we've, Eileen Hoffman has done some of that work, and she published the initial model in 2021. We're also trying to look at the larger scale detection, because with satellite surveillance, we get better coverage, but we don't always know what the species is. And so, yeah, and I think this was the wrong thing loaded, so I'm hoping there's not a gazillion slides here. But we still have trouble explaining everything and the interannual differences in the bloom magnitude. We know mixotrophy is important, and we're still trying to get a handle on just how mixotrophic it is in the model. Organisms have to be about 50%, obtain about 50% of their carbon heterotrophically in order to develop into blooms of the sizes we observe. But essentially, we've learned a lot, and we're trying to we continue to try to try to fill in these pieces. Sorry, I'm trying to talk fast. We got a late start. So we put numbers on a lot of these parameterizations that are now feeding some of our biological models. But there are emerging threats. I know some people have asked about those, and we still have a lot of unquantified nutrient sources. And if you were here at the meeting, we talked a lot about tidal flooding as being one source of nutrients that aren't being quantified in the restoration, groundwater inputs, as well as inputs from the sediments and sediment-rich suspension. We're also having warming seas, and we don't know if this is opening new niches for new species to emerge. We think it has, because we had Alexandria monoleum emerge in 2015, and perennial species were detected for the first time in a Bay program sample in 2018. And we have a paper about that, Womi et al., 2023. We just published that, and we actually had a bloom this year in 2023 of Cape Hap in the lower bay that we're just finishing up counts on. We've also been interrogating images in the offshore mid-Atlantic region, and we're finding a lot of different perennial species there as well. So I think there are emerging threats we need to worry about. But an exciting new program we're trying to do to enhance our monitoring and surveillance and to develop early detection of warning systems are to build and deploy planktoscopes, which are low-cost imaging systems that we can use to image everything in the water. People get the $150,000 IFCB or FlowCam if they can afford it, but these are much lower in cost, on the order of thousands of dollars. Again, we're going to use this surveillance and combine our surveillance at all different temporal and spatial scales to develop these forecast systems for the lower bay, which Dante talked about in his talk in our HAB session earlier, but unfortunately the people online didn't get to see that. But anyway, if anyone has questions, wants to be part of this network, we want to make it Chesapeake Bay wide. And right now we have funds we've deployed our first 8 planktoscopes. We have funds to deploy 22 more. And we're having a workshop next Friday to deploy the next 9 of them in the lower bay. And so we really would like to have people contributing images and developing essentially a bay-wide forecast for blooms. And then hopefully we can work on freshwater too. And with that, thanks. And we can get on to your questions.
Thanks a lot, Margie, and thanks to the whole panel. So we received some prior questions ahead of time, and I'm trying to summarize those. Believe there's still an option to submit questions via the chat. What's that? Oh, yeah, and then there's people in the room too. Okay, so I'm going to start with some of the pre-questions, and of course, these have been dealt with to some degree by the panelists, but I guess what we're looking for now is more your professional opinion type of things with some of these. So the first one is pretty hard. What would you say are the causes of HABs? And what can be done to prevent them? So we'll start with Dr. Kristova.
Oh, okay. So, okay, I think that we need to work at least 10 more years to be able to answer these questions, but with the basic HABs, we have this specific video blooms in very low nutrient systems, because that is the major understanding that the nutrients, nitrogen and phosphorus are the main factor which is influencing, stimulating the bloom. But with the benthic, we have cases in national parks, in rivers in national parks, in nitrogen limited conditions. Also, we run experiments with growing some microcoreas in complete lack of nitrogen. It survives for 40 days and producing toxins. So the nutrients are factor, but still we need really to work a little bit more on this to know what exactly is the effect of nutrients. Also, temperature in general, cyanobacteria, they like higher temperatures. So definitely temperature light intensity is associated with these conditions are favorable for cyanobacterial growth. And yeah, I would say these are the major factors. And of course, there are biotic factors, their interactions with other microorganisms also working on this question.
So, Dr. Randy?
Yeah, that's a good question. It's, you know, it's the 25 million.
Hang on.
Okay. Good question. And I think the answer to that is very species specific. Harmful algal blooms, you know, encompass very different organisms. The most common ones are dinoflagellates, which you know, Margie talked about some down in the lower bay, and they're what are called mixotrophic. So they can be autotrophic, as in they can photosynthesize, but they can also take up organic nutrients. So the different forms of nutrients that are coming into the bay have a different impact on the different types of harmful algal blooms. And so that has to be taken into account when we're considering harmful algal blooms. But the majority of them are dinoflagellates or cyanobacteria, which used to be called blue-green algae, although there's a rogue diatom as well, Pseudonychia. So we have to consider the species composition, but as Rosalina said, you know, climate change is definitely having an impact. It's particularly on cyanobacteria. They like warm temperature, but also dinoflagellates, because warming temperatures causes a stabilization of the water column, and dinoflagellates, which are motile, like that more than diatoms. And so I would say the two main things are probably the eutrophication, the input of both at this point, a lot of non-point sources of nutrients, as well as climate change. Those are probably the two biggest factors.
Yeah, and I'll add to the behaviors and biology of the dinoflagellates, because dinoflagellates are also connected to the sediments via their life stages. They form cysts that reside in the sediments, so we think that requires some sediment resuspension to get them initiated. And the other thing is estuary transport. Where blooms initiate is different from where they're transported to. And so the factors affecting their biology are different. And so really, if we hope to stop blooms, it's nice to study the initiation, the issues and the biological factors promoting their initiation. The other thing I would add with climate change is we're also finding climate change can have positive effects in terms of if we get bloom initiation in a shallow water tributary, then when it's too hot, it won't initiate. And so we can nip initiation in the bud at some of our initiation sites by the warming. It just gets too hot. And so, but if they're already initiated, once that heat comes, then they've been transported, so they continue to thrive. So.
Thank you, Margie. I think one of the things that needs to be emphasized, and it's becoming clear, is that when we use the term harmful algal blooms, HABs, we're not talking about the same thing. We're talking about something that has a similar impact, but we're dealing with vastly different habitats, like benthic in streams versus planktonic in dinos, but of course there is still an interaction, but so it's probably not one size fits all, I think. So we need to understand both the similarities and differences of different types of HABs. One of the questions that came up during the session was that we don't know as much as we'd like to about what causes the initiation of HABs. We can correlate variables we have a lot of data once the HABs have started, because it's obvious. But anyone who's tried, at any rate, I'm not going to linger on my experience, but and it seems to me like that might be one area where volunteer monitoring might be helpful to tip us off when something's going on, or adding some parameters to regular agency monitoring perhaps that might tip us off. So I'd like our panel to address how we might better deal with understanding and measuring initiation blooms. So let's start with Judy.
Yeah, that's again, the $25 million billion question. I do think that modeling is going to help us and forecast, and Peter Tango, who's here in the office, in the audience, worked on sort of nowcasting and forecasting microcystis blooms in the Potomac River. And I think coming up with ways to predict where when they're going to occur is really important. And that initiation stage is really hard. For instance, working on red tide and Crania brevis in the Gulf of Mexico, they still haven't found out where the cysts are, as opposed to the red tide in Maine, the Alexandria, where they know where the cyst beds are. So sort of tracking all those different parts of the bloom, and that initiation is, as Margie pointed out, is key. So we need to work on both finding that initiation phase as well as forecasting.
Yeah, and I'll add to what Judy said is one of the things that we did is we coupled some techniques in the lower bay in order to sort of do a reverse physical oceanography to track the bloom back. We joined forces with Jen Shen up at VIMS, who had a particle tracking model that we then could seed phytoplankton into different parts of the bay and watch how it's transported. Initially, we found it in the open estuary, but it all seemed to be coming out this pipe that we hadn't measured. And so we tracked it back inside to the Elizabeth and Lafayette River. And that's how we found that site of initiation where we could then, wrap our studies of bloom initiation around. So combining the models with measurements can be really useful. And we combined that with data flow observation. So again, it was a multi-scale operation. We also looked at the satellites because again, we could see where it would go after it initiated. And we've done this in several years since, because one time we had the bloom start 6 days before it got to the ocean front. And we were thinking, how did that happen? But when they ran the hydrodynamic model for that particular year, the hydrodynamics were just right to get that bloom up and around the bay and to the ocean front. In other years, there was a stationary eddy once it got into the James River and it just smeared up and down the river. So I think coupling multiple scales of technologies is going to be really useful in identifying those sites so we can understand the initiation.
And
Rosaline.
yeah, I would like to add quickly. So with the benthic cyanobacteria in streams, still we are in a very early stage of studying and understanding them. But there are two, I would say, major projects, and I'm involved in those. So we have an NSF project which study the seasonal dynamics of cyanobacterial blooms in Northern California, several streams. So the field team is collecting data from early in the season, let's say early spring until late fall, biotical data, and also a lot of abiotic measurements, which will be used for modeling. And here the project on Shenandoah River, which will provide a second year of data. It started last summer and it will continue, it's going on. This summer, this type of research will provide data about, hopefully answering this question, and the data are analyzed.
Okay, thank you. Okay. If you had to guess, what percentage of turbidity in the bay is due to phytoplankton versus suspended sediment? So, anyway, it's not a HAB specific question, but we'll go for it.
I can start there.
Yeah.
I think it depends on where you're at. In the shallow parts of the bay where you have bloom initiation, oftentimes in the lower bay, if you have wind-induced resuspension of the sediments, which can also cause nutrient inputs and cyst inputs into the water column, when you have that resuspension, you also have a lot of sediments. And so in that case, you're going to get a huge fraction of it, the turbidity being sediment. So, but then when you get to the more open water, the wind's not going to penetrate to the bottom. So I think your turbidity is much more likely going to be due more often to phytoplankton. I agree with Margie.
Other comments? Okay.
You know, I agree with Margie, and and that sediment component, when it's resuspended, can actually help stimulate the blooms, as we've seen in some of the talks today, and also, you know, on the work that I was doing up on that we are doing up on the Susquehanna Flats. Sediment seems to be very important in providing the phosphorus for the Microcera lingbia, because it's a nitrogen fixer. It tends to be growing in the low nitrogen areas, but it's getting the phosphorus from the sediment, and there's plenty of phosphorus in that sediment from the Susquehanna River. It's very fine grain, and it gets stuck on the filaments, and I said it like packs its lunch with it. It like sticks, the sediment sticks to the filaments, and it can float up, but it takes the sediment with you. So that doesn't exactly answer the question, but sediment's very important in that interaction with HABs.
Yeah, that is also true for streams, although the sediments usually they don't make the water turbid, it stays clear. You see the benthic algae on the bottom, and even if they're attached to hard substrate, let's say rocks, still there is a thin layer of sediments which play the same role. They just provide additional nutrients.
Yeah, and I'll add to that, we had a couple presentations in the session we had earlier that nitrogen is resuspended with the sediments, and that's important in the lower estuary. And then in some of the lakes and fresher water systems, phosphorus is released at high pHs, and phytoplankton increase the pH and you get phosphorus release. And when there's low DO, you also get phosphorus release. So the sediments are very connected. But the turbidity question is, you know, again, going to be depend on shallow deep water probably.
Yeah, just quickly, I'll throw in one item from my experience and correlates with Judy's. So we cleaned up many of the shallow areas of the bay. We had SAB coming in that decreased the amount of sediment in the water column and made it possible for light to reach the bottom in the littoral zone. And that's where Microcera, for example, hangs out. So it's complicated, right? And we'll just leave it at that. Let's see. Well, I wanted to go back just a second to, Margie talked about her planktoscope and how that is going to deploy to citizens, and that will hopefully be able to alert you to early stages of blooms and actual spatial distribution.
Yeah, it will actually initially, it's some of the uploading of data is really complicated. I went into more detail earlier that the workflow can be pretty complicated. So initially our deployments are with people on the water, people already making observations, people that have support systems that help them with running the sample is pretty simple. Getting the data uploaded and to where it needs to be is a little more complicated. And so we're trying to initially strategically deploy with people that have time to work with this. So there we have them deployed with some teachers, people at the aquarium, the Hampton Roads Sanitation District, the Department of Health, Department of Environmental Quality in Maryland, the Department of Natural Resources in Virginia. So, but then we hope we can eventually expand out.
I just want to make a comment about another program that EPA runs called Cyanoscope. It is tied into iNaturalist, and they even have kits that you can get. So I think that would be more relevant for the benthics, cyanobacteria. You go to your local stream, you grab some of whatever filaments are growing there, you bring it back, you take pictures. Those can be either identified with iNaturalist or sent to the planktoscope headquarters. And that could be another way of citizen involvement. It just seems to me like citizens are a way to really amplify our spatial knowledge.
Yeah.
And I'll just add that Maryland DNR Kath Wozniak and her group are trying to implement similar citizen science and other groups to implement similar HAB scope type instruments as well.
Yeah, I'd like to
Go ahead.
I'd like to add a note to the Cyanoscope. If I'm correct, that is EPA project, and I think that even they just give people free Cyanoscope. So it makes sense to get in contact with EPA. So it's probably option to get the microscope. It's small field microscope and it's really helpful.
Yeah, and I can give you directions on links for that.
And
Go ahead.
so what I wanted to say that the citizens are welcome, at least in my lab. I'm already receiving samples from people that are concerned, oh, is it dangerous? What is it science bacterium? My lab is open for very quick identifications.
Okay, let's see. Got time for maybe one or two more. Question from the in-person audience. What role does or can CBP play in HAB work? How do we incorporate into Beyond 2025 and NOAA CPBO 5-year plan?
I can start with that since I'm the salty one. There you go. And so one thing that we would love to do, we take in all the phytoplankton samples for the Chesapeake Bay program at the Phytoplankton Analysis Lab at ODU, at least the Virginia segments. And so one thing that we would like to do with the Bay program cruises is put our IFCB out during the cruises, and that would get us a lot more data, and we'd be able to match up with satellites, and that I think would greatly augment the Bay programs. Sampling, it would just get much better spatial coverage. It wouldn't improve the temporal coverage, but already during Bay program cruises, the staff will pick up samples when the chlorophyll hits a trigger on the underway system. So we'll get samples that come in to us then. And
So
so there's a.
Your machine can go out on the boat.
The IFCB can, and we're working actually to get the planktoscope, that lower cost version, onto a drone, essentially a hydrone or a blue boat. There is the company that makes them, they call them a blue boat. And so we're actually going to do a first deployment this summer where we plumb our planktoscope to be on a blue boat and run it with a drone.
All right, does anyone else want to comment?
I don't have anything more to really add to what Margie said, other than we really do need monitoring, and we need not just monitoring in the Chesapeake Bay, but we need monitoring in our forgotten bays of the Maryland coastal bays as well. So,
Rosalie?
We want to know what is in the rivers, right?
I would suggest that the Bay program could actually get behind some of this volunteer monitoring stuff and make the equipment as available as possible to interested parties, with the idea, of course, that they have to use it, and otherwise it'll come back. But I think that would really be helpful.
One other thing would be,
Go ahead.
we talked for years about trying to come up with a Chesapeake Bay HAB bulletin,
Mm.
and a coordinated group such as one of those two would be really helpful in trying to maybe piece that out. That pulls in not only the Chesapeake Bay photo monitoring, but any satellite data and the models that are being developed into a synthesis that could be.
Maybe volunteer.
And you would have this freshwater in the, mid bay and the southern.
And we have had task forces in both Maryland and Virginia already, so there already is some coordination, so that would be fabulous.
Okay, great. Let me see. We got it. Some of these are a little. Let's see. Or is anyone aware? I mean, I think we're all aware. But the question arises as to whether HABs are actually increasing over time, or whether we're just more sensitive to them and more observant. And are there any, so you can comment on that, or have there any HAB issues or occurrences that have actually been resolved so that they're no longer HABs, and what was done there? I can comment on the last part. The HAB blooms of microcystis in the Potomac River were in fact eliminated by advanced wastewater treatment, which basically cut the phosphorus. It took about 15 years though. That's a problem, is with some of these treatment mechanisms and some of these nutrient reduction strategies, there's lags in the system. So, and I suspect the same thing will be true with other HABs. But anybody want to comment?
Well, I think in terms of the cyanobacteria on Susquehanna Flats, I think, the magnitude of the blooms and, the duration having increased, that they weren't observed before. The species may have been there, but not in such large quantities. And we're also now seeing, as Margie alluded to, the emerging HABs. The publication, the Wolney et al., we described the emergent species of Karenia that are occurring, and we have 10 transects offshore, and there are hotspots of Karenia that are coming up of 3 different species, but they're also starting to come into the bay. So I think the evidence is that they are increasing, and we're getting a change in the emergent ones that are coming in with warmer waters.
Exactly. We're seeing a lot more come in, both not just in the Bay program samples, but in the shellfish monitoring that we do in Virginia is those samples come in monthly from all the growing areas around the state. So that is much better coverage than the Bay program phytoplankton monitoring. And we are seeing species emerge, as well as in Lake Anna, where we're doing the study, that is certainly a lake where the cyanoHABs have taken off, where they once were not. They are now a real problem.
Okay, we have about 1 minute left, and I want to throw it to Rosalina, because she has experience in California and a lot with stream HABs. So what's your
Very quickly, with the benthic stream cyanobacteria, we realized that there is an issue probably 10 or 15 years ago when the dogs reports on the dog kill started. So this is a very new research, so we don't have this historical data, we cannot compare. But that is indication that probably really now we have problem, and probably it was not like that 20 or 30 years ago.
Okay, well, I think our time has expired, and sorry about the glitches at the beginning, but I think I want to thank our panelists. Give them a hand. And I want to thank CRC for putting this on, because it's really an important issue. And I want to thank all of you, both in the room and at home. And of course, any of us is open to further discussion if you have points that you want to ask us via email. So with that, we will close the webinar.